Hey guys, so I decided to get back into making biology videos. Anyway, I just wanted to finish up the chapter at least, in case I get in trouble. So concept 5.4, proteins include a diversity of structures resulting in a wide range of functions. Nearly every dynamic function of a living being depends on proteins. In fact, the importance of proteins is underscored by their name, which comes from the Greek word proteos, meaning first or primary. Proteins account for more than 50% of the dry mass of most cells, and they're instrumental in almost everything organisms do. Some proteins speed up chemical reactions, while others play a role in defense, storage, transport, cellular communication, movement, or structural support. Figure 5.13 shows examples of proteins with these functions, which you'll learn more about in later chapters. So here we have figure 5.13, enzymatic proteins. We have enzymatic proteins, defensive proteins, storage proteins, transport proteins, hormonal proteins, receptor proteins, contractile and motor proteins, and structural proteins. Life would not be possible without enzymes, most of which are proteins. Enzymatic proteins regulate metabolism by acting as catalysts, chemical agents that selectively speed up chemical reactions without being consumed in the reaction. Because an enzyme can perform its function over and over again, these molecules can be thought of as workhorses that keep cells running by carrying out the processes of life. A human has tens of thousands of different proteins, each with a specific structure and function. Proteins, in fact, are the most structurally sophisticated molecules known. Consistent with their diverse functions, they vary extensively in structure, and each type of protein having a unique three-dimensional shape. Proteins are all constructed from the same set of 20 amino acids, linked in unbranched polymers. The bond between amino acids is called a peptide bond, so a polymer of amino acids is called a polypeptide. A protein is a biologically functional molecule made up of one or more polypeptides, each folded and coiled into specific three-dimensional structure. Okay, so for my notes, I wrote, most functions of a living being depends on proteins, speeds up reactions, defense, storage, transport, cellular communication, movement, structure. Most enzymes are proteins. Regulates metabolism by acting as catalysts, which are chemical agents that selectively speed up chemical reactions without being consumed in the process, and enzymes can be used repeatedly. Proteins are constructed from the same set of 20 amino acids linked in unbranched polymers. Peptide bonds are bonds between amino acids, so a polymer of amino acids equals a polypeptide. Protein is a biologically functional molecule made up of one or more polypeptides, each folded and coiled into a specific three-dimensional structure. Okay, so amino acids, monomers. All amino acids share a common structure. An amino acid is an organic molecule with both an amino group and a carboxyl group. The small figure shows the general formula for an amino acid. At the center of the amino acid is an asymmetric carbon atom called the alpha carbon. Its four different partners are in an amino group: a carboxyl group, a hydrogen atom, and a variable group symbolized by R. The R group, also called the side chain, differs with each amino acid. The R group may be as simple as a hydrogen atom, or it may be a carbon skeleton with various functional groups attached. The physical and chemical properties of the side chain determine the unique characteristics of a particular amino acid. thus affecting its functional role as a polypeptide. So here we see this is the alpha carbon, the hydrogen atom, the carboxyl group, the amino group, and the R group. Figure 514 shows the 20 amino acids that cells use to build their thousands of proteins. Here, the amino groups and carboxyl groups are all depicted in ionized form, the way they usually exist at the pH found in a cell. The amino acids are grouped according to the properties of their side chain. One group consists of amino acids with nonpolar side chains, which are hydrophobic. Another group consists of amino acids with polar side chains, which are hydrophilic. Acidic amino acids have side chains that are generally negative in charge due to the presence of a carboxyl group, which is usually dissociated ionized at cellular pH. Basic amino acids have amino groups in their side chain that are generally positive in charge. The terms acidic and basic in this context refer only to the groups in the side chains because all amino acids, as monomers, have carboxyl groups and amino groups. Because they are charged, acidic and basic side chains are also hydrophilic. So this is the graph showing you all of the 20 amino acids. There are only 20 amino acids, and these build up proteins. These are essentially the alphabet for proteins. Okay, so here for my notes for amino acids, I wrote an amino acid is an organic molecule with both an amino group and a carboxyl group. Alpha carbon is an asymmetric carbon found at the center of the amino acid. Its four partners include an amino group, carboxyl group, hydrogen atom, and a variable group symbolized by R. R group or side chain can be simple or complex, such as one hydrogen atom as the R group or side chain, or a carbon skeleton with functional groups attached as the R group or side chain. Okay? Characteristics of an amino acid depends on the physical and chemical properties of the side chain, or R group, thus affecting its functional role in a polypeptide. There are 20 amino acids that cells use to build proteins, group according to their properties of their side chain. Nonpolar side chains are hydrophobic, polar side chains are hydrophilic. Amino acids have side chains that are generally negative in charge. Basic amino acids have amino groups in their side chains that are generally positive in charge. Since both acidic and basic side chains are charged, they are hydrophilic. Okay. Polypeptides, amino acid polymers. Now that we have examined amino acids, let's see how they are linked to form polymers. When two amino acids are positioned so that the carboxyl group of one is adjacent to the amino group of the other, they can become joined by a dehydration reaction with the removal of a water molecule. The resulting covalent bond is called a peptide bond. Repeated over and over, this process yields a polypeptide, a polymer of many amino acids linked by peptide bonds. You'll learn more about how cells synthesize polypeptides in 17.4. Okay. So this is a chart of how they make a polypeptide. The repeating sequence of atoms highlighted in purple in 515 is called a peptide backbone. Extending from this backbone are the different side chains, R groups, of the amino acids. Polypeptides range in length from a few amino acids to 1,000 or more. Each specific polypeptide has a unique linear sequence of amino acids. Note that one end of the polypeptide chain has a free amino group, the N-terminus of the polypeptide, while the opposite end has a free carboxyl group, the C-terminus. The chemical nature of the molecule as a whole is determined by the kind and sequence of the chains, which determine how a polypeptide folds and thus its final shape and the chemical characteristics. The immense variety of polypeptides in nature illustrates an important concept introduced earlier, that cells can make many different polymers by linking a limited set of monomers into diverse sequences. So for polypeptides, amino acid polymers, I wrote, when two amino acids are positioned in a way so that the carboxyl group of one is adjacent or opposite to the amino group of another, they can attach through dehydration reaction, which is a removal of a water molecule. The resulting covalent bond is called a peptide bond. The repeating sequence of linking amino acids by peptide bonds results in a polypeptide backbone, to which amino acid side chains, R groups, are attached. One end of the polypeptide has a free amino group, the N-terminus of the polypeptide, while the opposite end has a free carboxyl group, the C-terminus. Chemical nature is determined by the kind and sequence of the side chain, which determine how a polypeptide folds, and thus its final shape and chemical characteristics. I see. Protein structure and function. The specific activities of proteins result from their intricate three-dimensional architecture, the simplest level of which is a sequence of their amino acids. What can the amino acid sequence of a polypeptide tell us about the three-dimensional structure, commonly referred to as simply as the structure, of the protein and its function? The term polypeptide is not synonymous with the term protein. Even for a protein consisting of a single polypeptide, the relationship is somewhat analogous to that between a long strand of yarn and a sweater of a particular size and shape that can be knitted from the yarn. A functional protein is not just a polypeptide chain, but one or more polypeptides precisely twisted and folded and coiled into a molecule of a unique shape, which can be shown in several different types of models. And it is the amino acid sequence of each polypeptide that determines what three-dimensional structure the protein will have under normal cellular conditions. So we have the different models and diagrams in this figure. We're not going to go over that. When a cell synthesizes a polypeptide, the chain may fold spontaneously, assuming the functional structure for that protein. This folding is driven and reinforced by the formation of various bonds between parts of the chain, which in turn depends on the sequence of amino acids. Many proteins are roughly spherical, globular proteins, while others are shaped like long fibrous proteins. Even within these broad categories, countless variations exist. A protein-specific structure determines how it works. In almost every case, the function of a protein depends on its ability to recognize and bind to some other molecule. In an especially striking example of the marriage of form and function, Figure 517 shows the exact match of shape between an antibody, a protein in the body, and the particular foreign substance on a flu virus that the antibody binds to and marks for destruction. Also, you may recall another example of molecules with matching shapes from Figure 2.3, endorphin molecules produced by the body and morphine molecules, a manufactured drug, both of which fit into the receptor proteins of the surface of the brain cells in humans, producing euphoria and relieving pain. Morphine, heroin, and other opiate drugs are able to mimic endorphins because they all have a shape similar to that of endorphins, and can thus fit into and bind to endorphin receptors in the brain. This fit is very specific, something like a handshake. The endorphin receptor, like other receptor molecules, is a protein. The function of a protein, for instance, the ability of a receptor protein to bind to a particular pain-relieving signaling molecule, is an emergent property resulting from exquisite molecular order. Okay? And something to note is that I just want to make the connection that, when you exercise and move in general, any kind of movement produces endorphins, right? So movement equals endorphins, which is synonymous with morphine, heroin, and opiate drugs. So if you want to produce euphoria and pain relief, you can just, even a little bit of movement will release endorphins that will fit into your brain cells, and it'll have similar effect, even if it's just a little bit, a fraction of a degree of change, right, guys? Okay. So for protein structure and function, I wrote, activities of proteins depend on their three-dimensional structure, which is the sequence of their amino acids. Polypeptide and protein are not synonymous. Yarn versus sweater. A functional protein may consist of one or more polypeptides precisely twisted, folded, and coiled into a molecule of unique shape. The amino acid sequence of a polypeptide determines what structure the protein will have under normal cellular conditions. The folding of a polypeptide is driven and reinforced by the formation of bonds between parts of the chain which depend on sequence. Globular, spherical proteins versus fibrous, long fiber protein. Just, know the difference between globular and fibrous. A protein-specific structure determines its ability to recognize and bind to some other molecule, which results in a function. And endorphin molecules and morphine molecules both fit the receptor proteins on the surface of brain cells. Morphine, heroin, and other opiate drugs have similar shapes to endorphins. Okay. The 4 levels of protein structure. In spite of their great diversity, proteins have 3 superimposed levels of structure, known as primary structure, secondary structure, and tertiary structure. A 4th level, quaternary structure, arises when proteins consist of 2 or more polypeptide chains. Figure 518 describes these 4 levels of protein structure. Be sure to study this figure thoroughly before going on to the next section. So this is the chart. Okay. Primary structure is a linear chain of amino acids. I'm just going to go over this briefly because I don't want to read the entire text per se, But primary structure is a linear chain of amino acids. Secondary structure depends on regions stabilized by hydrogen bonds between atoms of a polypeptide bond. Tertiary structure results in... this is the process. Okay, I'm sorry. Okay, yes. Primary structure is the linear chain of amino acids. So this is just the yarn itself, right? In secondary structure, the regions become stabilized by hydrogen bonds between atoms of the polypeptide bond. So this is when the yarn starts to take some form of a shape through hydrogen bonds. within the different molecules in the yarn shape. Tertiary structure is the three-dimensional shape stabilized by interactions between side chains. So in secondary structure, we have the hydrogen bonds between the actual atoms of the polypeptide bone. And just remember, the side chains are the aspects of the polypeptide bone that are sticking out. So the tertiary structure is when there are bonds between the side chains rather than the backbone itself. And quaternary structure is when there are two or more polypeptides. This applies for only some molecules. And one example of a quaternary structure would be hemoglobin, which is an oxygen-binding protein of red blood cells. Okay? So, 4 levels of protein structure. Primary structure, the actual sequence of amino acids, the order of letters in a very long word. Sequences are determined by inherited genetic information and dictates secondary structure and tertiary structure. Secondary structure, coils and folds that are the result of hydrogen bonds between repeating atoms of the polypeptide backbone. Examples include helix and beta-pleated sheet. Tertiary structure, interactions between the side chains, R groups of the various amino acids, and determines the overall shape of the polypeptide. Hydrophobic interactions. An example, hydrophobic interactions and disulfide bridges, which is in the chart. You'll be able to understand that, I promise. Quaternary structure, 2+ polypeptide chains, some proteins. This is the overall protein structure that results from the aggregation of these polypeptide subunits. Transthyretin has 4 polypeptides, collagen has 3 polypeptides, and hemoglobin has 4 polypeptides. Okay. Sickle cell disease, a change in primary structure. Even a slight change in primary structure can affect the protein shape and ability to function. For instance, sickle cell disease, an inherited blood disorder, is caused by the substitution of one amino acid, valine, for the normal one, glutamic acid, at the position of the 6th amino acid in the primary structure of hemoglobin, the protein that carries oxygen in red blood cells. Normal red blood cells are disc-shaped, but in sickle cell disease, the abnormal hemoglobin molecules tend to aggregate into chains, deforming some of the cells into a sickle shape, which is like a crescent. A person with the disease has periodic sickle cell crises, when the angular cells clog tiny blood cells, impeding blood flow. The toll taken on such patients is a dramatic example of how a simple change in protein structure can have devastating effects on protein function. So, just for reference, this is a normal red blood cell, and this is a sickle cell blood cell. So usually this can kind of like move about within the blood cells easily, but this can get clogged up. This can get stuck and clogged in the vessel, causing sickle cell crises. Okay, so for sickle cell disease, I wrote a change in primary structure. Slight changes in primary structure can affect the protein shape and ability to function. Sickle cell disease, which is an inherited blood disorder. Okay? Substitution of one amino acid, valine, for one glutamic acid at the 6th amino acid position for hemoglobin. Normal red blood cells are disc-shaped, but the substitution results in a sickle shape. Sickle cell crises are when the sickle-shaped cells block blood vessels, impeding blood flow. What determines protein structure? You've learned that a unique shape endows each protein with a specific function. But what are the key factors determining protein structure? You already know most of the answer. A polypeptide chain of a given amino acid sequence can be arranged into three-dimensional shape determined by the interactions responsible for secondary and tertiary structure. This folding normally occurs as the protein is being synthesized in the crowded environment within a cell, aided by other proteins. However, protein structure also depends on the physical and chemical conditions of a protein's environment. If the pH, salt concentration, temperature, or other aspects of its environment are altered, the weak chemical bonds and interactions within a protein may be destroyed, causing the protein to unravel and lose its native shape, a change called denaturation. Because it is misshapen, the denatured protein is biologically inactive. Most proteins become denatured if they are transferred from an aqueous environment to a nonpolar solvent, such as ether or chloroform. The polypeptide chain refolds so that its hydrophobic regions face outward towards the solvent. Other denaturation agents include chemicals that disrupt the hydrogen bonds, ionic bonds, and disulfide bridges that maintain the protein's shape. Denaturation can also result from excessive heat, which agitates the polypeptide chain enough to overpower weak interactions that stabilize the structure. The white of an egg becomes opaque during cooking because the denatured proteins are insoluble and solidify. This also explains why excessively high fevers can be fatal. Proteins in the blood can tend to denature at very high body temperatures. A protein in a test tube solution has been denatured by heat or chemicals. When a protein in a test tube solution has been denatured by heat or chemicals, it can sometimes return to its functional shape when the denaturing agent is removed. Sometimes this is not possible. For example, a fried egg will not become liquefied when placed back into the refrigerator. We can conclude that the information for building specific shape is intrinsic to the protein's primary structure. This is often the case for small proteins. The sequence of amino acids determines the protein's shape, where an alpha helix can form, where beta-pleated sheets can exist, where disulfide bridges are located, where ionic bonds can form, and so on. But how does protein folding occur in the cell? Okay. So what determines protein structure? My notes here says a polypeptide chain of a given amino acid sequence can be arranged into a three-dimensional shape determined by the interactions responsible for secondary and tertiary structure. Also depends on physical and chemical conditions of the protein's environment. pH, cell concentration, temperature. Weak chemical bonds may be destroyed, unraveling the protein. Denaturation. Most proteins become denatured if they are transferred from an aqueous solution to a nonpolar solvent, chemicals disrupt the hydrogen bonds and ionic bonds and disulfide bridges that maintain the protein's shape, or excessive heat that overpowers the weak interactions that stabilize the structure. High fevers can be fatal. Proteins in a test tube solution can return to its functional shape when the denaturing agent is removed. The sequence of amino acids determines the protein's shape. Protein folding in the cell. Biochemists now know the protein acid sequence for over 100 million proteins, with several million attached each month, in three-dimensional shape for tens of thousands. Researchers have tried to correlate the primary structure of many proteins with their three-dimensional shape to discover the rules of protein folding. Unfortunately, however, the protein folding process is not that simple. Most proteins probably undergo several intermediate structures on their way to a stable shape, and looking at a mature structure does not reveal the stages of folding required to achieve that form. However, biochemists have developed methods for tracking a protein through such stages and learning more about this important process. Misfolding of polypeptide in cells is a serious problem that has come under increasing scrutiny by medical researchers. Many diseases such as cystic fibrosis, Alzheimer's, Parkinson's, and mad cow disease are associated with the accumulation of misfolded proteins. In fact, misfolded proteins of the transthyretin protein featured in 518 have been implicated in several diseases, including one form of senile dementia. Even when scientists have a correctly folded protein in hand, determining its exact three-dimensional structure is not simple. For a single protein has thousands of atoms. The method most commonly used to determine the 3D structure of a protein is X-ray crystallography, which depends on the diffraction of an X-ray beam by the atoms of a crystallized molecule. Using this technique, scientists can build a 3D model that shows the exact position of every atom in a protein molecule. Nuclear magnetic resonance spectrography, cyro-electron microscopy, and bioinformatics are complementary approaches to understanding protein structure and function. The structure of some proteins is difficult to determine for a simple reason. A growing body of biochemical research has revealed that a significant number of proteins, or regions of proteins, do not have a distinct 3D structure until they interact with a target protein or other molecule. Their flexibility and indefinite structure are important for their function, which may require binding with different targets at different times. These proteins, which account for 20 to 30% of mammalian proteins, are called intrinsically disordered proteins and are the focus of current research. Okay. So for protein folding, my notes, I wrote, researchers know the amino acid sequences for over 100 million proteins. Researchers have tried to correlate primary structure with their three-dimensional shape to understand the rules of protein folding, but it's not that simple. Proteins go through many intermediate structures on their way to a stable shape. There are many stages. Misfolding of polypeptides in a cell is a serious problem, which can result in cystic fibrosis, Alzheimer's, Parkinson's, and mad cow disease. Determining exact 3D shape is not easy due to thousands of atoms. Most commonly used method to determine 3D structures of a protein is X-ray crystallography, allowing scientists to show the exact position of every atom in a protein molecule. There's also nuclear magnetic resonance, spectroscopy, cyro-electron microscopy, and bioinformatics. Intrinsically disordered proteins are proteins that do not have a 3D shape until they interact with a target protein or molecule. Okay? So that's it for concept 5.4. Next video will be concept 5.5. Nucleic acids store, transmit, and help express hereditary information. Okay? So I hope this helps you guys. I'll see you guys next time. Have a good day. Bye-bye.